Muhammad Umar Farooq Biological video. Please make sure to subscribe, like and share. Ask the question for answer, and you can also comment to each other for solve the problem as well. So here is we will talk about the mechanism of starch, protein and fat synthesis in the plant. So how we produce the starch, fat and protein in the sense of fruit, grains or something. So now we will discuss about the whole story of the synthesis and their biochemistry. So this is the lesson about the biochemistry. So today we will discuss here. So that is the plant cell which that is contain a chloroplast. Chloroplast is very important organelle to produce sugar by using the light ATP. Using the light to produce ATP in the thylakoid membrane. So here is the thylakoid which that is produce the ATP, NADPH and basically NADPH and ATP. So here is the thylakoid membrane which that transport the ATP and NADPH into the stroma, and fixation of the carbon dioxide. The fixation of the carbon dioxide produce produce a sugar molecule, 3 carbon containing sugar molecules in the Calvin cycle, and release the oxygen from the light reaction. So important things here is you can say that suppose it is the plastid, and the plastid is different type. Here is the chloroplast, oleoplast and proteoplast, or some chromoplast. So we will discuss is the detail in the detail. So here is the G3P which that is 3 carbon containing with phosphate group attached to further process by the glucose synthase, fructose synthase enzyme to use and that's produce sucrose. So basically sucrose it will be produced, and these sucrose convert into the simple sugar, and this simple sugar go into the conversion by the glycolysis by the single molecule to produce pyruvate 3 carbon containing molecule, and these 3 carbon containing molecule will be produced 4 molecules will be produced from the 2 sugar molecules. So that is go into the mitochondria and attached by the to produce acetyl CoA, and this acetyl CoA will produce here we will discuss about the pyruvate. So here is the 3 carbon containing molecule which that's attached enzyme CoA, and this produce acetyl CoA, and this acetyl CoA will be attached in the Krebs cycle by the oxaloacetate to produce citrate, and that citric acid cycle will begin to produce FADH2, NADPH and ATP. So ATP and FADH and NADPH will be produced, and that is go into the electron transport chain. And so here you can say that here is the oxaloacetate which that convert into the citrate by the fixation of acetyl CoA, which that is 3 carbon containing, and 4 basically oxaloacetate is the 5 carbon, and 1 carbon molecule will be attached. So that's convert into the 6 molecule citrate, and that's 1 carbon dioxide will be released, and here is the production of FADH2, NADPH2 and ATP. This content is the energy production, energy using, and electron donor, proton donor, basically electron donor which that's donate the electron in the electron transport chain. So electron transport chain produce more ATP, but on the other hand this is the energy beside the starch, protein and fat synthesis. So now we will discuss about here is you can say that the acetyl CoA will export even and produce fat by the malonyl CoA conversion by the HMG CoA reductase enzyme. So it's a long discussion, but here we will discuss about the main component of the synthesis of this content. So here I am drawing the plant which that contain leaves and roots. Root attach the tubers, potatoes tubers, solanum tubersum, On the other hand, the fruit in the form of, for example, suppose it's not, but suppose olive in case of olive plant, so that is olive contain a oleoplast because fat reserved into the oleoplast, it is known as oleoplast. But here we will discuss about the cross section of the potatoes which that contain it is also known as tubers. So these contain fibers, a thick fiber which that is part of the fiber, and there's contain tubers contain a fiber of cells which there's a fiber like structure. Its mean is the oleoplast. So remember it, and this oleoplast is the fiber of cells. And if we will discuss and separate the one fiber or one cell of the amyloplast, basically it's amyloplast. Fat using is the oleoplast, but starch storage is in the amyloplast. It is also known as plastid. So this nucleus and plastid, which that here is the nucleus and here is a cell which contain a large granules of the amyloplast. So that is the storage of the amylose and amylopectin. Anyhow, it's not more discussion about it. So we will get soon the uh, uh, absorb the concept of the synthesis of protein and fats, and this whole story will be wind out by the concept, and we can uh, learn the chemical reaction. So we will discuss about here. So outside here is the cell which contain a plasma membrane. So the outside is the sucrose or glucose. So GLUT5 or GLUT1 channels are present in the cytoplasm, so cell membrane. So this channel will allow to sucrose inside or glucose inside by the active transport. It means the 3 sodium molecule will be go inside to allow the sucrose inside by using the ATP, and that is also known as active transport because using the energy is not a passive transport. Passive transport is using for the diffusion or without ATP due to the osmotic gradient. So here is the sucrose or glucose will enter into the cytoplasm. So is the cytosol. Inside of the cytosol, sucrose basically sucrose or glucose is enter. So we will discuss about the whole concept. So here is the water will be absorbed to basically water absorption to uh, sucrase enzyme used to produce glucose and fructose. So this sucrase enzyme produce glucose and fructose. So these fructose, if we will talk about the on the other hand, here is the glucose simple, which that's convert into glyceraldehyde 6 phosphate by the using of enzyme dehydrogen phosphatase enzyme. So this conversion of the dehydrogenase glyceraldehyde 6 phosphate dehydrogenase enzyme used to add the phosphate group on the carbon number 6. So here is the fructose 6 phosphate which that is also produced from the here is the fructose by the using of NAD plus to NADH basically dehydrogenase enzyme by the inorganic phosphate group at H. So here is the F6P convert into the again dehydrogenase enzyme same reaction to produce 1,6 bisphosphocructose or fructose 1,6 bisphosphate. So two phosphate group attached, carbon number 1 and carbon number 6 contain a phosphate group. So remember it, that is the reaction is known as glycolysis. So is the glyceraldehyde and dihydroacetaldehyde which that's produced, and this can be reversed by the production of NAD plus to reduce is known as reduction is NADH or dehydrogenase to produce again its reversible reaction also. So 1,3 bisphosphoglyceraldehyde will be produced, and this go into the further ADP convert into ATP, and these glucokinase produce a 3-phosphoglyceraldehyde, mean 3 carbon number 3 contain a phosphate group, and that is a phosphoglucomutase enzyme used to convert the 2-phosphoglyceraldehyde. Carbon number 2 contain a phosphate group. So that is important, and here is the enolase enzyme used to produce a PEP, phosphoenol pyruvate, and this go into convert a pyruvate by the chemical reaction, and that is also reversible. So that's go into the mitochondria by the transporter, and that's acetyl CoA will be produced, and that's acetyl CoA will be go outside or used to Krebs cycle, and that's when go into the outside acetyl CoA, which that produce on the presence of HMG CoA reductase enzyme to produce malonyl, and then malonyl go into the produce a fatty acid and into the oleoplast. So that is the reaction is also in the oleoplast, but proteoplast is also known. But here we will discuss about the plastids. The collectively we are collectively understanding the plastids because I am not want to draw separately. So that's all proteoplast, oleoplast, and amyloplast is in the same short or single short. So here is the phosphoenol pyruvate can be enter inside of the plastid, and that's produce a pyruvate by the releasing of inorganic phosphate, and this pyruvate will be CoA absorb or react and produce acetyl CoA, and this acetyl CoA will be released into the cytosol, and that's produced by the malonyl CoA on the presence of HMG CoA reductase enzyme to produce CoA to produce a fatty acid which that is reserved into the seed, and acetyl CoA is again will be attached by the acetyl malonyl convert into the malonyl CoA to that's production and reserve of the fat into the seed is in the oleoplast is this is the oleoplast is the reserve for the fat in the form of lipids or triacyl glycerol and fats. So major is the unsaturated fat are present into the seed is common. So here is the 3-PGA from here to move inside also, and that's conferred, and that's chemically PGM phosphoglucomutase enzyme to produce and 2-PGA, and that's our reaction is look like a glycolysis, and this will be inside and go into the plastic to produce from here 3-PGA or so on the or something other region. So these 3-PGA will be produced by the adding of ammonium ion to produce serine, glycine, acetyl, serine, cysteine, methionine. That is the amino acid, and this amino acid will transport into the basically that is the plastic. We will talk about the proteoplast, so that's store the seed into the seed or something which that is the protein reserve by this chemical reaction from 3-PGA to a fixation of ammonium ion. So if we will talk about from here, that is if enter into the transporter of the plastid glyceraldehyde dihydroacetone phosphate or glyceraldehyde phosphate, which that is reduced by the NADH to produce NAD plus, and that's known as dehydrogenase enzyme to produce 1,3-bisphosphoglyceraldehyde phosphate. 1,3-bisphosphoglyceraldehyde. So this is the production, and that is convert again, that's down the chemical reaction to produce again protein or fat. So if we will talk about from here, so that is the important things which that glucose 6-phosphate will enter into the transporter channel, and that's go glucose 6-phosphate to convert fructose 6-phosphate and that's 6-phosphate isomerase enzyme to produce, and that's a dehydrogenase enzyme to produce a fructose 1,6 bisphosphate, and these bisphosphate will be produced a two molecules again look like a same in the glycolysis. So that's if we will talk about the upward moving, that's enzyme used to fixation starch synthase enzyme. It's a long story about the chemical reaction from here. So that's is here is a basically proteoplast, and here is the oleoplast, which the reserve, and here is the amyloplast. If we will talk about, that's amylose will be stored into the plastid. So that's our reaction is done here.